In a previous lesson, we asked the question, why do memories undergo the consolidation process? Why are they transformed over time? Why are they reorganized across multiple brain sites over time? And we considered three possibilities. Interference due to new learning within the hippocampus. So it may be that new learning can interfere with previously learned information. After all, there are a finite number of synapses in the hippocampus. We had interference due to new neurons. So new neurons, as they take place within the hippocampal circuitry, may be interfering with previously established memory traces. And then we had the interference due to synaptic resetting during sleep. So while we learn things during the course of the day, synapses get stronger. But in order to prepare the hippocampus for new learning on the next day, we need to weaken those synapses back to some baseline level, and that may be happening during sleep. Well, that would interfere with previously established memory traces. So in general then, memory consolidations may be protecting a memory from potentially disruptive changes in the hippocampus. In this lesson, we'll see if we can figure out how the brain does this consolidation process. And some insights into this process have emerged from the study of hippocampal place cells. Now, place cells fire when the animal occupies a specific location within its environment. And in this diagram, we see a mouse that's going to be running this pathway here. And the colored dots represent a particular place neuron firing in response to the location of the rat. So as the rat is in this location, there is one particular place cell that shows lots of activity. All the little pink dots would be action potentials by that place cell. As the rat moves on in the environment, a different place cell starts to fire. As the rat continues its journey, the colors here represent activity of different place cells. And so you see a given place cell is only responding at a certain location. As the rat moves to a new location, a different place cell responds. In a moment here, we'll take a look at a video of this process. Okay, so here we have a maze-like contraption. The rat is down here, and there are electrodes inserted into the hippocampal region, recording from different place cells. And as the rat moves through this environment, different place cells will become active, and you'll see that record over here in this graph here. Okay, so again, the idea is as the rat moves through the environment, different cells become responsive. And these are hippocampal place cells. Now here we can see sort of a record of individual place cell activity over time, recording from cells number 1, 2, 3, 4, 5. So for example, in the beginning of the rat's journey, it is cell number 1 that shows maximal activity. And as the rat proceeds through the environment, cell number 2 becomes active, and then cell number 3 becomes active, and then 4 and 5, etc. So the place cells are responding to the location of the animal in its environment. Now let's suppose that this rat was moving through its environment and then got a reward. It might want to remember the journey that it just took. So the sequence of place cell activity represents the movement of the rat through the environment. And if the rat is going to remember its journey, the sequence of place cell activity would be part of that memory trace. Now, if we take a look at this diagram here, we have a population of the hippocampal cells that fired in some sequence during the animal's journey through the environment. Now, let's say that that journey was eventful. There was a reward at the end of the journey, so the animal may want to remember its journey. Well, the idea is we've seen that some of the same cells that were involved in processing the initial episode are going to be involved in a memory trace of that episode. So consolidation then is this transformation of the memory, the strengthening of a memory. Now, if we think that it involves some of the same cells involved in processing the episode, then the consolidation process must be activating those same cells somehow. But this presents a problem, because we want to reactivate those cells to strengthen the connections, but we want to do so without behavioral repetition. We don't want the animal, as it's consolidating the memory, to constantly relive the memory. So memory consolidation must be detached from behavior in some way. Otherwise, memory processing would interfere with the processing of the current situation. So how does consolidation occur? How does it occur in a way that doesn't interfere with the animal's ongoing experiences? A clue emerged when scientists continued recording after the animal went through its environment. So the record up here shows the initial pattern of place cell activity as the animal moved through the environment over time. Cell 1 showed activity here, and then cell 2, 3, 4, 5, etc. And then after the animal was sort of finished exploring its environment, they recorded the following pattern, this down here, and they called it a forward replay. What they found was a compressed version of the same sequence of place cell activity. This occurred right after training. So the animal might just be resting a moment after it made it through the environment, and they recorded, notice the time scale here, 250 milliseconds versus 8 seconds here that it took to initially go through the environment. But we see the same pattern, cell 1, 2, 3, 4, and 5, just as we did in the original exploration of the environment. So we have a replay of the sequence of place cells. It's compressed in time. And scientists suspect that the replay may create the necessary conditions for synaptic plasticity in those cortical neurons that will store the memory for the event. In other words, the replay may be an important part of the consolidation process. So back to this diagram, if the initial encoding of the experience involved this sequential activity of 1, 2, 3, 4, and 5, the replay is reactivating those cells in a compressed time frame, and that may be helping to strengthen the connections between the neurons involved in making a memory. Now, one way to test whether replay is involved in memory consolidation is to disrupt replay. So after an animal goes through an environment and has learned something about that environment, scientists have disrupted this replay, prevented the replay from happening. And disrupting replay can impair memory for recently learned tasks. So many researchers argue that some memory consolidation processes occur during sleep. There's lots of evidence that sleep enhances memory consolidation. As it happens, replay is present during sleep, and that further suggests its role in consolidation. So to help us visualize what's going on, here we see the hippocampus here and the cortex here, and we'll say that system consolidation, again, is that reorganization of a memory over time. And it may be that the hippocampal replay phenomenon is part of the process of consolidation. So we have some synapses that were strengthened during some learning episode during the day, and right after that learning episode and during sleep, the hippocampus may be replaying in a compressed fashion, replaying that activity in order to facilitate synaptic plasticity in cortical neurons. So this is the nature of the consolidation process, that the replay phenomenon is setting up the conditions for plasticity to help the cortex strengthen the cortical aspect of the memory trace. And this then is a reorganization of the memory over time, and replay may be an important part of that process. Now, the idea that sleep helps consolidate memory is, of course, important for anybody interested in how best to learn anything in life. And so, from a student's point of view, when we're learning things during the day, we definitely don't want to disrupt sleep or not sleep if we want to remember the information later on. So sleep is a critical part of consolidation of memories, and therefore becomes an important part of any effective study strategy. So why do this consolidation during sleep in the first place? One idea is that this eliminates any interference due to ongoing perceptual processing. You'll recall that when a person is asleep, they're no longer having perceptual experiences, and this allows then the cortex to be engaged by the replay phenomenon, so the cortex can do memory consolidating without the interference of ongoing perceptual experience. Now let's consider some other observations about this forward replay. Retrieving relevant memories is a crucial function of a memory system. Sensory input is a cue to memory retrieval. Forward replay often occurs when a task has a choice point. Now, the replay happens in advance of the actual choice point, as if the animal is retrieving relevant memories to guide decision making. So forward replay may also be a marker of memory retrieval, not just consolidation. So consider the mouse or the rat down here. We have a T maze, and let's say the reward is at location B. Now the mouse has already sort of explored this T maze, right? So it's already now made a memory that the reward is located at choice at option B here. So it has to make a right turn. And the idea is when the mouse is in this part of the chamber, scientists have recorded from these place cells, they've recorded sequential patterns of activity in certain place cells. And it's as if the animal is reactivating a memory of what happens when you go left, what happens when you go right. So the replay may be an indicator that the animal is retrieving previous memories. And the memory would involve a certain sequence of place cells when it turned right, a different sequence of place cell activity when it turned left. As it happens, the turning right sequence of place cells led to the reward. So as the animal approaches the choice point, it's going to be retrieving memories of what happened in the past when it went left, what happened when it went right, and it can use that retrieval of memory to guide ongoing behavior. In other experiments, scientists have recorded from place cells in environment 1 and then put the animal in environment 2. And sometimes they will record a replay of the place cell activity from environment 1. And this is interesting. So replay from environment 1 can occur in environment 2, and this overlap might be how the brain links environments and events. And a given place cell could be part of the replay for both environments. So when the animal's in environment 2, it may be reactivating a memory of environment 1 because there might be some relationship between environment 1 and environment 2. And so by reactivating these memories, the animal's brain may be linking these memories for events in environment 1 and 2. So in summary then, replay may be a mechanism for both consolidation and retrieval. And notice then, this means that consolidation of memory begins early. It happens soon after the experience. So replay is most prevalent immediately after the experience and decays to chance after about 24 hours. And if replay is part of consolidation, then it means the consolidation of memory is happening soon after the event itself. It's just that we might not be sort of conscious or aware of these sort of replay events taking place in our brain. The intensity of replay during pauses, but not prolonged activity, predicts later memory. So scientists have found that the more replay and the more intense that replay, that's correlated to better memory for whatever was just learned. And replay has been detected during sleep, which is known to contribute to memory consolidation. So hippocampal replay may be one of the mechanisms for how the brain is consolidating memories.